Thanks, Neil. And for those of you who don't know, Jonathan's the star of a TV show called Creepy Crawlies. You should, am I right, Creepy Crawlies?
Am I?
I vaguely recall seeing you somewhere on there. I'm Sujevaan Ratnasingham from the Biodiversity Institute of Ontario. And I'm going to be covering two projects, so I'm just going to run through it and try and make the time, probably won't. They're fairly complementary projects, the Barcode of Life data systems and the International Barcode of Life project. Oops. The Barcode of Life data systems is an informatics platform for the barcoding community. This is a research community that's focused on generating a reference library of gene markers that can be used to identify species. They follow a strict metadata standard and loci standard as well. And so they use this platform to collect, analyze, and publish the data. The platform is more than just a database. It stores the data, it stores the primary data, it houses workflow tools, is backed by computational capacity so that researchers in developing nations can perform their analyses without having to have the infrastructure. I won't go into much further detail than that. It's online at www.boldsystems.org. The other project is the International Barcode of Life project. This is a 26-nation project, started in 2010, is being run for 5 years, has a very specific goal to barcode 5 million specimens from across the world and collect the ability to identify 500,000 species. This is a huge advance in our ability to recognize species on the planet. There are two numbers we keep in mind. One is 1.8 million. That's the number of described species so far. And the latest is 8.7 million, which is the number of estimated species on the planet. Now, I am talking about eukaryotic life, so the prokaryote interests won't, I guess you can look at your laptops for a little while. This is a fairly wide project because of national involvement. So the funding models are complex, but the governing model has been simplified. In the past, when we started off, we had a massive steering committee of 80 individuals, but have since streamlined it. There's a single board composed or led by Genome Canada in Canada, which will pull in funding, members of funding agencies. So it's very much funder-driven. They're informed by a research oversight committee that wants to make sure that we're staying on task, but also that the products that we produce are scientifically relevant. It's led by a scientific director, project manager, and an international scientific steering committee. This scientific steering committee only consists of those individuals that are putting in large sums of money into this project, either through sampling efforts, sequencing capacity, or meetings and other infrastructure-related items. The history of this project is fairly lengthy, going back to 2003 with the original publication of the Hibert et al. Paper on barcoding. And since then, we've been lucky enough to have funding agency after funding agency drop in and give us millions of dollars. So far to date, about $50 million accumulated, starting with the Moore Foundation. And a lot of Canadian agencies. Genome Canada has been the latest primary driver of this effort. A couple of dates that are critical. The Barcode of Life data systems launched in 2005. A data standard was established by the Consortium for the Barcode of Life following in that year, and the IBOL project launched in 2010. This seems to have been a very successful approach where the informatics platform appeared first, which allowed the standards community to look at the data that was being collected, very much in the same workflow that GSC has followed. and then establish standards based on that data. In fact, we're in the process of reviewing the standard and revising now that we are approaching 2 million records. The IBOL project launched after reaching 1 million records, where it was clear that there was global interest in this and the system worked. The assets that we have as a result of this project now in IBOL and also in the Barcode of Life initiative are labs. There are labs around the world that are now dedicated to DNA barcoding at the University of Guelph, the Canadian Centre for DNA Barcoding, the Kunming Biodiversity Institute, at the Smithsonian, the Natural History Museum, and they range in size. For example, the CCDB in Canada can produce about 500,000, process about 500,000 samples a year, whereas the one at the Natural History Museum can handle about 10,000 samples a year. And it's important to have these different sizes to support varying projects. We also have a large network of sampling groups and sampling programs, 3 represented here. Over at the top is our mobile vehicle in North America that goes around to fixed sites, collecting samples on a periodic basis. So in a sense, mobile observatories, if you will, because they hit the same sites. We hit museums, bottom left-hand corner, and on the right-hand bottom is a barcode survey supported by Toyota. Over the years, we've been collecting a large amount of data, geographically diverse, but I do want to point out a few areas. So there are actually genomic observatories or compatible sites here. 3 of them are highlighted. One in Costa Rica, over 100,000 samples collected from a research park there. Churchill, of course, is highlighted there. And in Guelph, we've been collecting about 80,000 samples. Melbourne. There are a few others because this infrastructure is in place. These are great for expanding as goes. We're collecting eukaryotes. These GO sites or potential GO sites can be mini in nature. Again, mostly built for eukaryotic sampling. These are 2 sites close together. They're monitored every summer repeatedly at the same time to look at the impact of urbanization in southern Ontario. Shared interests. I think our greatest benefit is a community of scientists that have a passion for this work, for collecting samples, a massive amount of samples, and follow strict protocols after almost 9 years of this kind of work. A bioinformatics platform designed for handling eukaryotic life, access to field sites that are used for macroorganism sampling but can easily be extended for microorganism sampling. Most of it is terrestrial, but there are a couple of marine. What we can gain is extended metadata standards, time series genomic data to help contextualize our survey plans. We still have a long way to go, and we need to get a feedback loop in place, ecosystem models, and also potential GO sites. So I'll just stop there.